Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Advances in HPAPI Development and Regulatory Strategy. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Aerogen Life Sciences who developed the content for this presentation. Aerogen Life Sciences is a leading R&D and manufacturing solutions provider for the life sciences industry worldwide. It offers end-to-end integrated and standalone solutions for small molecules and biologics from early discovery to commercial manufacturing. Established in 2001, the company operates through a network of sites locally or located globally with a team of over 4,500 scientists and 450 plus PhDs. Its expertise and experience have enabled over 400 customers to advance their research programs from discovery through commercialization. Aerogen's innovative mindset, infrastructure, flexible business models have enabled them to serve large pharma, biotech, agrochemical, animal health, and performance chemical industries globally. Now, it is my pleasure to welcome our speaker for today's event, and he is Dr. Sankepa Rai. He's a Senior Director of Technical Operations with Chemical Development and Manufacturing Solutions at Aerogen. So Sankepa is going to join us with his webcam on shortly, and there he is. Hi, hello there, Sankepa.
Hello, Sonia.
Sankepa is a seasoned pharmaceutical professional with over 2 decades of experience in small molecule discovery, process development, CMC, HPAPI, ADCs, and contract manufacturing. Currently serving as Senior Director of CMC Technical Operations at Aerogen Life Sciences, leading end-to-end CMC development and late-phase programs for NCEs, APIs, and intermediates. At Aerogen, he is responsible for process development, validation, commercial supply, along with P&L ownership for chemical development solutions. He plays a strategic role in customer engagement and long-term collaboration building. Now, with the introductions out of the way, now it's my pleasure to pass the mic and the controls over to Sankepa. So Sankepa, when you're ready, you may begin.
Thank you, Sonia, for a nice introduction. Hello, everyone. Good evening, good morning. Thank you for joining. Let me take you through quickly on a topic, or I should say my favorite topic, advances in HPAPI development and regulatory strategy. In next 30 to 40 minutes, I'm going to talk about what we do at Aerogen, how high potent compounds playing an important role in modern day drug development, and how is it impacting the patient's life throughout the world, and how Aerogen can help, and what are the control strategies, what are the challenges we face, what are the regulatory strategies which we may have to follow, what are the controls regulators have put in. Those are the topic I'm going to talk about. Before that, I just want to take a minute to add on to what Sonia said about Aerogen. At Aerogen, we all believe in a purpose, or in every molecule is a possibility of better health. Either it is for animals or humans, or it is for birds. So this purpose motivates each and every employee in this company to collaborate with our clients and bring their molecule to market. So ultimately, it's going to impact some lives across the world. So that's the purpose which drives all of us. In next 30 minutes, I'm going to talk a little bit about what are HPAPIs, what sort of molecules, which forms HPAPIs, what is the current market trends and the growth drivers, what are the advantages of HPAPI development, advances in terms of the technology, considering the latest AI revolution, regulatory landscape and strategy, and also the future outlook. And later on, I'm going to take a few questions from your end, whatever I could answer on this topic. So these are the points I'm going to cover next 30 minutes. First of all, let me quickly talk a little bit about what are HPAPIs. HPAPIs are highly potent active pharmaceutical ingredients. Okay, when we call it as highly potent, means this has got very high efficacy in its therapeutic area. For example, if I consider a medicinal chemist doing a research, finding a molecule for one specific therapeutic area, if he gets an nanomolar compound, he gets highly excited because I got a nanomolar compound. That means it has got a high efficacy on a specific therapeutic area. Similarly, high potents are those kind of compounds which are discovered with high efficacy for any specific therapeutic area, and we name it as high potent active pharmaceutical ingredients, which goes into making the formulated products. This also shows activity in lower doses. The advantage of lower dose is like you don't have to take a 500 mg or 600 mg tablet just like a Dolo 650 mg what you take. You can take a 10 mg tablet once a day or twice a day. It will show its desired activity. And what it does is it shows very less side effects on human body and it shows more of a therapeutic activity. These high potent compounds, either they can be cytotoxic or it can be cytostatic in nature. So I'll talk a little bit more about what are cytotoxics, what are cytostatics in nature in the upcoming slides. And I will also quickly touch upon how important is high potent compounds in modern day drug development or the developing a new drug for the new diseases which are impacting the lives across the world. So what I know is historically, probably if you consider 15, 20 years back, way back in 2000, people used to dose highly potent compound as such to the human being. Of course, it used to act on certain therapeutic areas, but it used to give more of a side effects than its actual therapeutic activity. So that is the reason people have come up with a lot of new targeted therapies or a precision, we call it as precision medicines. Few of them are ADCs, PROTACs, and many others which are in the pipeline, which are going to come in. Majorly, the applications of high potent compounds are used in oncology or cancer drugs. It's not limited to oncology. This also has got applications in hormonal therapies, pain management, cardiovascular drugs. So these high potent compounds plays a very vital role in all these therapeutic areas. Now, having said that, he found a molecule which is highly active at lower doses. There is always association of some challenges. Either it may be handling, manufacturing, and of course dosing. One of the major challenges, like as soon as the compound is highly active at lower doses, you can't expose yourself to that compound yourself, even not on the nanogram scale, because that will have impact on your body or a human body. So the important part there comes is a containment. How do you contain? How do you operate and manufacture it? How do you do the packaging? How do you do all the logistics? Containment is one challenge where people have come up with various kind of isolators, glove boxes. Then you have to operate in front of those containers, you always have a opportunity that you'll expose one or the other ways to those drugs. So that is when you have to use PPEs, personal protecting equipment, when you handle those drugs. Other challenges like how do you design? You just can't make a box just which is closed, which is not open to anywhere you can't access to. Those kind of designs will have a challenges in operation. So that is where our lot of chemical engineers came up with the facility design, which is easy to operate in terms of isolators or glove boxes, and that also should avoid cross contamination. So the challenge here is if this compound contaminates to any other product, the impact is going to be huge. So the facility design should be such that it is easy to operate and it avoids any cross contamination to any other products. Similarly, the other challenge is analytical. You need to test quality of the product, you need to release them. So when I say that, you also need to have analytical setup, which is within the isolators. The other major challenge is the waste which is generated. You can't just dispose it because that will have certain percentage of your product which will have impact to the environment around you. So waste disposal is another challenge. Of course, there are solutions which I'm going to discuss in the subsequent slides. Regulatory requirement, regulators become very alert when you're handling a highly potent compound, and it also has some impact to the operations, how you make that drug. Those are the challenges of handling a high potent compound. I'm going to talk a little bit more about those in details in the subsequent slides. Sonia, before I do that, I have a poll question to our audience. If you can flash that.
There you go. Okay, audience members, this is our first poll question. This is done in real time, so your participation is strongly encouraged and very much appreciated. So the question we have for you is, what percentage of currently marketed drugs are high potent? So you have a couple of options below to choose from. So go ahead now and make your selection. Is it A, 35%, or is it 41%? 25%, and then the other option we have is 10%. So please go ahead and make your selection. I'll leave this open for another 20 seconds or so just to get everyone involved in this poll question. Then I will close the polls, share the results, and then Sankepa will give us the answer. Okay, Sankepa, that looks like it's the majority of our audience. There's still a few more people out there that have not made their selection, so I'll leaving it open for a few more seconds to get everyone involved. Okay, that looks like it is everyone. Thank you so much, everyone, for participating in poll question number 1. Now let's close the polls and take a look at the results. Okay, so here are the results, Sankepa. 35% said 47. Or sorry, 47% said 35, 12% said 41, 24% said 25, and 18% of our audience said 10%. So Ken Sankepa, what is the correct answer?
Sure. Thank you, audience, for your poll and participation. So, yes, the 10% as expected is a majority, but the right answer is the 12% of audience who said 41%. Uh, because the reason being, if you look at the current market, whatever the drugs which are available, either it may be onco product or non-onco products, you will see highly potent compound. So the right answer is 41%, and I see in the coming years it's going to increase above 50%. Reason being, like I mentioned in the previous slide, the precision medicines, targeted therapies, the new technologies to deliver your drug to a specific area. So that makes the high potent compound is as one of the targeted medicine for the future drugs. So that's the reason it's going to increase moving forward. But thank you for participating in that poll. I'll quickly go on to the next slide. Uh, yes, this slide talks about how do you band or how do you categorize your molecule into various bands. Uh, like I said, as mentioned in this slide, OEL banding is subjective to each organization. But what we need to look at is the OEL value. When you say what is OEL value, occupational exposure limit. For when you do a banding, you calculate the OEL for a period of 8 hours. So universally, when the exposure is more than 500 grams per meter cube over a period of 8 hours, it is categorized as a OEB1. It can be handled, which is categorized as a low, expected to have a low toxicity, and it can be handled in an open, general ventilation. OEP2, where the exposure limit is 100 to 500 micrograms per meter cube, where this has to be handled with the LEVs or with the partial containment, which is called as OEP2. Any molecules which has got a OEL level 1 to 100, that's where the activity or the potency starts dropping, and we need to start having more and more controls in handling. That's called as OEB3, OEB4, any compound which has got at a exposure limit of 1 to zero point 0.1. It's OEB4, that's where you need full containment, isolators, RTPs, and OEB5, where you need to have maximum containment, PPEs, dedicated facilities. Most of the compounds which falls into OEB5 are cytotoxic or hormones, but not all of them. Like I said, I repeat, OEL banding is subjective. You can categorize OEB3A, 3B, and you can divide the OEL ranges. Each organization does it differently. At Aragen, we have OEB6. We categorize into OEB6. Anything less than zero point 0.1 micrograms per meter cube, we call it as OEB6. Compounds, and we take special precautions to handle those. I'll talk a little bit about it in the subsequent slides. Before I do that, why do people develop HPAPIs? How does the market trend look like? If you look at the what is the total global market from 2022 to 2027, it's expected to grow from 24.5 billion to 39.6 billion by 2027 with a CAGR of 10.1%. If you look at the small biotechs which are developing HPAPIs, which is around 1.3 billion. Currently, it is around zero point 0.8 billion in the HPAPI market. When I consider what is the opportunity for outsourcing, there is a huge potential for the CDMOs to grab these development opportunities, provided you have better control, containment, and compliance within the organization. So this quickly about the brief market outlook, which looks very positive. Like I said earlier, moving forward, HPAPIs, targeted therapies, are going to be the medicines for the people around the world. Having said that, we also need to understand what are the advances currently is being made to handle high potent compounds, how to reduce exposure, how to improve the efficiency. Those are the things which is currently being done uh, around the world, and one of them is continuous manufacturing. The advantage of continuous manufacturing is you just do the manufacturing or synthesis. You don't isolate, or you limit the isolation steps. Take the molecules forward. Once your synthesis is done, if you have to do a crystallization, take the stream for crystallization. Isolate the solid, use it for blending, do the granulation, sizing, do the tablet, press, coating, packaging, and isolate your product as a tablet or any other dosage form. It's very easier said than done. There are regulators who will be looking after how are you going to have a control either in your synthesis or on the crystallization or in the blending. The important part is we need to have a controls. You have a samples which is taken out in a controlled way, in a contained way, test it, take it forward, and isolate in as a tablets or any dosage form as a final product. What it does, is it reduces exposure to the human who's manufacturing, and also it improves efficiency. It also helps us to scale the drug to the higher level, and also it helps us to meet the market demand in a much faster way. The other advances which is being made currently is microdosing. Normally, when you develop a drug, first thing what you do is the toxicity test, then you go for efficacy. When you do the efficacy, you also try to find the dose range finding. That's where you dose the drug at various ranges and try to find what is the toxicity as well as efficacy and its ADME properties. Current trend is you do a microdosing at a very lower level, since it is a high potent compound, and establish its efficacy and figure out what happens if you dose it higher levels without dosing a species on a higher dosage. So that's another advances which is being made in the modern day drug development, especially in high potent compound, because if you dose any animal to the higher extent, it may just die off. So that's another advances which is being made currently across the world. Uh, One other point I just want to talk about advances, continuous manufacturing I briefly covered, microdosing I covered. The other advances is the technology which is being developed to contain the manufacturing of these high potent compounds. Uh, the containment has developed various technologies to have a robust and safe handling of high potent compounds. Few of the things is like it's very simple. You just segregate the hazardous waste and non-hazardous waste. When I say that, how do you define what is hazardous and non-hazardous? You just can't say that. You have to test it and figure out whether your high potent compound is there in your waste or not. And other thing is having a proper airflow which controls the micro, just to give a proper ventilation to the people who work around that facility, and have appropriate construction or a construction design for the facility where you have this containment. The other thing is cleaning and decontamination protocols. I'll talk a little bit more about it because which is very important part when you develop a high potent compound. We need to have a proper cleaning procedure for the equipment what you use, the downstream equipment what you use, the PPEs what you use, and how do you clean the surroundings, and proper decontamination protocols. Normally, the decontamination protocol is developed by a chemist which has developed this drug. The simple strategy is you degrade your final drug into a pieces where it doesn't show its activity at all. Normally, it is either oxidize it, reduce it. You use normally use sodium hypochlorite or some permanganate solution which is being used. Based on the developed process, either you may have to keep it for 8 hours all the way up to 24 hours. Then draw the sample, test it for your API content, then prove that your API is completely decomposed. That's the decontamination process. Once you are decontaminated, you just remove the waste, then you start cleaning your equipment. Once equipment is cleaned, check the cleaning sample for your main product and prove that your high potent compound is less than the limit based on the macro calculation. So that's the cleaning and decontamination procedures. Other thing is establishment of different zones based on the contamination risk. Like you may wet area, dry area, or maybe reactor area. You define and categorize different zones and have different control strategies. There are modern HVAC systems which prevents cross contamination, HEPA filters which filters the air and gives you the desired quality of air because you're making a high potent compound or a finished drug in your facility. I also see a lot of automation and coming into this manufacturing. I'll talk a little bit more about that in the subsequent slide. If possible, ideally use one single use containment system for your manufacturing so that you don't have to clean it repeatedly and use that facility for the entire manufacturing. Enhanced monitoring and control system, you have a DCS. You can sit in your AC room and look at your reactor. How is it behaving? Is there any challenges or any issues in your manufacturing batch? There is also a lot of focus on sustainability and energy efficiency, which I'm going to talk in subsequent slides. Before I do that, just a full question, Sonia, over to you.
Okay, no problem with that. Here is our poll question. Everyone, you should see it now appear on your screen. And there it is. Are all the oncology drugs high potent? So you have only two options here, yes or no. So just like we did in the last one, just like we did with our last poll question, just go right ahead and make your selection, and then I'll leave it open for about 45 seconds to get everyone involved, and then we'll close the polls, and just like we did before, I'll share the results and then get Senkepa's thoughts or get the answer from Senkepa. Okay, Senkepa, that looks like the majority of our audience, but we have a few more people making their selection, so I'll keep it open for a few more seconds. Okay, that looks like it's everyone. Thank you so much, everyone, for participating. Now, let's take a look at the results. The polls are now closed. Okay, so in our audience, 20% said yes, while 80% said no. So thank you very much, everyone, for that. Senkepa, what is the correct answer?
Yes, you are right. All the oncology drug need not be high potent. You're right, you're spot on, because there are lot different ways where your oncology drug acts nowadays. There are kinase inhibitors, there are KRAS. It was just that the targeted therapies which carries the high potent payloads. However, it goes into a specific area where in your body where your cells are impacted because of cancer. Thank you for participation. I will also take a moment to talk about cytotoxics and cytostatics. Cytotoxic drugs which will have impact on your gene, which will modify your gene. It will kill your cells. So those are called as cytotoxic drugs, which has to be handled separately, should have separate containment, separate facility to handle them. So with that, I'll go to a next slide. Before I do that, I spoke briefly about the containment, what are the technologies or advances which is being used. I just want to take a minute to show a video of what we have at Aragen in terms of handling high potent compound, both for development as well as manufacturing under GMP conditions, and how the world-class containment facility is being built. Thank you. I'll just show you a video for a minute. Thank you for your patience. This is going to be a world-class facility which we are currently validating. The facility is ready. We are validating the facility where you can do the discovery work, development, and GMP manufacturing. We can also do uh, linker toxin conjugation and conjugation with MAPs in this facility. Going to the next topic, which I wanted to touch upon, is how does the regulatory landscape look like? The major regulators, FDA or your local body, they all look at you when you do the drug development, especially high potent drug development. The ultimate goal is to ensure the patient safety, control the product quality, and avoid any legal activities, facilitating global market access to everyone. That's where regulators play an important role, and ensuring people follow a good manufacturing practices, starting from development, manufacturing, commercialization, as well as the life cycle management. The other focus area is contaminations, cross contamination, decontamination of high potent products. That's the area which regulators look at, and the important activity is the cleaning validation, cleaning method, and the testing method, and how is it validated, and how did you categorize exposure limits based on therapeutic area and dosage, and what sort of a risk-based approach you have been following, is all the risk covered, what sort of documentation which is being done, that those things are verified by the regulators and by periodic inspections and audits. When it comes to the regulatory trends in outsourcing and CDMO partnership, it's no different from an inventor company requirement versus the CDMO requirement. The quality and compliance requirement, safety remains the same. The CDMO, which follows the compliance, good manufacturing practices, are sustainable CDMOs, and Aragen is one of them. One of the important part is the stringent safety protocols. Like I said, decontamination, disposal procedures for high potent compounds, and provide the proof to the regulator transparently. Say this is the procedure what we follow. That's important for any CDMOs. Work safety, workplace safety, containment, environmental controls. These are the important part of any CDMO who is supporting the inventor companies for their uh, product development. Other approach what CDMOs follow is the risk management. That is important part of any activity, identifying the potential risk. This risk could be related to regulatory, compliance, product quality, or supply chain disruptions. We need to identify these risks and provide a mitigation plan for these risks as a CDMO. Implementation of mitigation strategies and ensuring continuous monitoring and controlling of your manufacturing process and supply chain process for the continuous supply of high potent drugs to the market. Coming to the strategy considerations, what are the points which we need to consider? Engage the regulator in the early stage of development. Arrange a meeting, identify the risk, and present it. Okay, this is what I'm developing. This is a potential risk, and this is what I want to do just to mitigate the risk. And this is the output which is expected. Have the risk mitigation strategy presented. Engage the regulator openly in a transparent way. Get their guidance, buy it, and make that strategy for the speeding of your high potent drug development. The other important part is how do you achieve the quality, right? So quality is subjective. uh, you just can't get the quality what you want until you design a process which gives you a desired quality consistently. So that is why we follow quality by design. It's just like any other drug development. You just figure out what sort of quality you need for your drug and figure out what are the quality attributes in your process, either it's a material, process, or environment. Do the risk assessment, evaluate the high risk items, design experiments. We have a software or tool called Design Expert. you can use either full factorial or a half factorial experiment and figure out a design space for operating your process. Have a proper control strategy, have specifications, testing methods which are validated, and look for continuous improvement in your process and testing methods. Important part is how do you manage the life cycle of your highly potent asset, I would say. Once you commercialize your product, there is always opportunity for continuous learning. When you do 20 batches, 30 batches in your manufacturing, you will get a data, put that into AI, and that will give you the optimum condition. Assume that you get a better process which gives you a better quality and yield. How do you implement that change? Provide proper justification to the regulator, do the supplementary validation, do that and implement the change. Formulation, packaging, labeling, there is another area you can always continuously improve, and any changes can be implemented with proper documentation, ensuring the compliance. So these are the strategies which are needed when you develop a high potent asset and manage it and help the patients across the world. How does the future look like? Now, when I see around Aragen, when I look at the sample management facility, I see a robot managing your compound, moving your sample from one desk to other desk. So that's where we are moving. Emerging technologies, AI-based drug design. I remember tens and thousands of drugs being samples or molecules being made before you get one active molecule. Now AI gives you the precise drug or scaffold where you get your hit. There are tools which help you to optimize the process. You don't have to do what if studies. You just put the reaction conditions, which it will tell you what works, what doesn't work. Automated manufacturing, which I spoke about, and also AI in formulation. You provide the structure, dosage, and the targeted therapy, which will tell you what sort of formulation will best suit for this drug. That's where the world is moving, and we are catching up with that. Other important part is sustainability. We need to reduce the waste for our next generation. Green chemistry philosophy: reduce the waste, have a zero discharge, try to have minimum waste from any of your reaction. One of the ways to do continuous manufacturing, there are new platform technologies like flow chemistry, where you can just have a small room, provide and manufacture tons and tons of drugs. Biocatalysis is another area which reduces waste. That's where the world is moving when it comes to the drug development. Uh, Similarly, the expectation is regulators keep up to the speed. There are ways to fast track review and approvals when you have a drug which is going to have a large impact to the patients. That's how our future looks like. That was my last slide. Open to questions. Sonia, over to you. Thank you. That was my brief about high potent drugs and its important in modern day.
So I just want to say again, thank you very much, everyone, for listening to this great presentation by Sankepa. Now we're going to begin the Q&A portion of the webinar. And remember, if you are watching this via LinkedIn live events, you can send in your questions using the comments window. And if you're watching this via GoToWebinar, use that questions window also in the control panel. Okay, all right, so let's start with this first question here. Sankepa, while you're speaking, I did receive a couple of questions that came in from our audience. So let's start with this first one here. This audience member is asking, what are Origin current capabilities to handle HPAPIs? Great question. What are your thoughts on that, Sankepa?
Yeah, thank you, audience. I briefly presented a video what we have at Aragen. We can handle molecules from OEB4 all the way up to OEB6. When I say OEB6, it's up to 10 nanograms per meter cube over a period of 8 hours OEA level, both on the discovery scale, development, as well as under the GMP conditions. We also have a capabilities to develop these toxins, normally OEP6 compounds, which goes and acts as the toxins for ADCs, which is a precision medicine. Do the conjugation. We can do that from milligram scale all the way up to kilogram scale at Aragen. To answer precisely, we can handle compounds with OEB level starting from 4 all the way up to 6. Thank you, Sonia.
Great. Thank you very much for that, Sankapa. Let's go to our next question. This audience member is asking, can you use all the high potent drugs as a warhead in ADCs? What are your thoughts on that, Sankapa?
Uh, Answer is no, because your high potent drug, when you conjugate with your MAP, with the linker, first thing, it should be stable. You should be able to achieve a certain DAR. Most importantly, when this high potent compound is delivered to a place where its activity is expected, it should show the activity and it should be stable. Some of the high potent compound does not stable are not stable at a specific pH. So not all of them can be the toxins, but majority of them yes, but mostly they are up to OEB6.
Okay, thank you very much. Okay, this looks like is our last question here. Is all of the HPAPIs are cytotoxic? What are the normal decontamination strategies for HPAPIs?
Thank you, Sonia. So uh, all the high potent compounds are not cytotoxics.
Mhm.
They can be cytostatic as well. There are drugs which are highly potent, but they are not cytotoxic. There are drugs which are used for cardiovascular uh, diseases, pain management. They are not cytotoxic. They, they cannot be killing your cells. So the answer is no. Uh, they are not cytotoxic. They can be cytostatic, and it can have different mode of action as well. And quickly on the contamination, I spoke about it. Just decompose your final product so that it loses its activity. Then test it, degrade it, dispose it. That is the way we decontamination. Decomposition, testing, and disposal are the three very, very important points or requirements when you do high potent drug development in your facility.
Okay, thank you very much for that answer. I'm just checking to see if there's any more questions coming in. Okay, all right. Thank you very much. That looks like it's the end of the Q&A portion of the webinar. Well, thank you very much for those answers. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks to gain access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, give me a few seconds. I'm going to share with you a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And I'll put that in the chat box right now. And there you go, everyone. You should see it there. Now, for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. And I'll go here to this slide here. Now, we mentioned that there is a LinkedIn link here for you to utilize to follow Aragen, and I'm going to put the LinkedIn right here for you to utilize and make it easy for you to access to it. Okay. Now, please join us in thanking our speaker, Sankapa, for his time here today. So thank you very much, Sankapa.
Thank you. My pleasure. Thank you, everyone.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone.